It must be like church, everybody's sitting in the back.
Welcome to the Reflections program this afternoon. You got through the rainstorm to get here. We appreciate that. It's appropriate today. Our program kind of remembers Earth Day, which we had last Saturday, but our speaker today, Erik, has spent his whole life involved in plant pathology and improving the crop yields and protection for small grains and corn. It's going to be, it's a little bit different, and it's
And
not
soybeans too, and soybeans as well.
Yeah, totally different, he said. But he brought his A-team. When I walked in, Verlin was up here setting everything up, so the two of them, many of you know, I was raised in Arkansas. Now, half of the state of Arkansas is flat and it's covered in row crop. And to me, row crop means beans and cotton and rice. And I spent 50 years in the state of Arkansas. I worked in the flatlands. And after talking with Erik some, I didn't know anything about production farming. I'm really serious about how it was done, the dollar impact, the impact on the communities there. All I knew is that cotton, you picked it and you wore it, and rice, you ate it, and beans, you sent to China to turn into edamame or something. And a quick personal story, even though it's Erik's day. When I was just starting in the construction business, my boss sent me to West Helena, Arkansas. Now, that is in the flatlands, and you have got to want to go there. But the locals, it was the fall of the year, the crops were coming up, and the locals would gather in the paved yard or the gravel yard at the cotton gin every morning early, and they'd tell tales and spit and whittle and whatever they did. Well, I wanted to fit in, so I walked from our temporary construction shed over to where all these guys were gathered around the back of a pickup, and there was a lull in the conversation, and I said to them, looks like that cotton's coming on good. Well, one of the old timers in his overalls, and he had about 5 days of beard and a little skull dripping down here, and he says, "Son, you was raised in the city, wasn't you?" Well, I said, "Yes, sir, I was, up in Little Rock." And he said, "I could tell." He said, "That's beans, that ain't cotton." Erik would have known better, and so my research prompted my visiting with Erik. He may cover some of this, but I thought it was interesting. My research prompted me to look a little bit. Agriculture and related industries in the United States accounts for $1 trillion of the gross national product. That's little over 5% of our gross national product. Now that was related industry like food prep and so forth. American farms themselves account for about $170 billion and 1% of our gross domestic product. The U.S. is the largest exporter of feed grains in the world, and they produce some 420 million tons of feed grains. Amazing stuff that I didn't know, and we're going to learn about how this man made a contribution to that part of our world. Erik, welcome. It's all yours.
Thank you, Buster. And I want to thank him also for preparing the slides for me, digitizing them. I had a whole collection of slides, much of which are gone when we moved. All my wheat work is gone, most of it. But anyhow, I'll talk to you a little bit about The way I started anyway, I've always been interested in plants. And when I was a teenager, middle school and high school, I used to mow neighbors' lawns. But what I would do, the guy that lived behind me worked for Northrop King and Company, and he would give me the old, the a-year-old seeds. I built a cold frame, and in peat moss pots, I grew up plants, and I helped landscape my clients' yards. And that was, one of the things I had done. And I knew I wanted to go into plant science. I went to the University of California at Riverside, and I got a degree in plant science. Actually, I did research in plant pathology, but they didn't award a degree in plant pathology. One of the things I worked on there was a bacterium that caused galls or cancer-like cells in carrot roots. And it was work that was sponsored by the American Cancer Society to find out what the tumor-inducing principle was. So that was my first start. And then I graduated from there, and I started graduate school at Oregon State University, and just went home for lunch before my physical chemistry exam, and there was my draft notice. So 2 years later, I came back. But anyway, there I worked on a vascular wilt of tomatoes. And there we discovered what we call a phytoalexin. It was a compound produced in response to the infection of the fungus. And the student that followed me actually identified the compound, but I identified how it started. In the resistant one, it came on very fast. In the susceptible variety, it wasn't there until the plant was dying. And so then I finished graduate school, and I took my first job at the University of Minnesota with the U.S. Department of Agriculture. And so some of the stuff I'm going to show you is from Minnesota. And so it's kind of a detective story, because many of these things we wonder how they got there. When I was at Minnesota, we divided the state up into 36 square mile quadrants, and I traveled all there looking at corn, soybeans, and small grains. And in the 4.5 years I was there, I found 9 new pathogens that hadn't been described. So it was fun. So I'll start here with, this is my family, Blacksburg, and my daughter is a chiropractor in Ohio, and my middle son is an emergency room doctor here in Roanoke, and a toxicologist, and my older son is a biostatistician for Medtronics. And, whoops, got the wrong one here. This is what I want. This one, he worked in my program, during the summer, and decided he didn't want to be a plant pathologist. He worked a lot. He was fun. And then he actually got a master's degree in plant pathology at the University of Minnesota. And he took his first job with the U.S. Department of Agriculture down in New Orleans, working in cotton. And then they lost their statistician. So he started doing the statistics for the folks there, the scientists there. So he decided he went back to school, and that's when he got a degree in biostatistics. And I tell you, it makes him a much better biostatistician having a degree in a biological science as well as math. And he's done very well. He's a senior biostatistician for them. So I guess that's enough about my family for now. What I want to talk about is, in order to have disease, you need to have the pathogen, obviously, but you need to have a favorable environment, and you need to have a host plant that's susceptible, or at least not highly resistant. And the pathogen can vary in its virulence, how virulent it is to cause disease. And the environment, the more favorable the environment, the easier for the disease to develop. So this I want to show you here is a field of sweet corn. And when I was out there, the corn is stunted, and there's a virus that got it. And I'll show you the next picture there. And that's what happens to it. The interesting thing is, I was able to determine that corn produces about 2 leaves a week during the summer. And so you could count back and figure out when did the infection take place, because this infection is systemic. Only the new growth shows the symptoms. And the importance of sweet corn was, or why sweet corn was so affected, was that the sweet corn processing companies stagger the planting so they keep the business working. They don't overwhelm the processing plant. And that really, whereas I think the next picture I'll show you here, yeah, there you can see why it's called maize mosaic virus. And this is a field corn leaf. You can see it's not quite as affected, but these were leaves that were higher up because the field corn was planted much earlier in the year. When I counted back the leaves, we determined that infection took place on the 2nd or 3rd of July of 1977. And that was all the way across the places where I found these, the leaves I could count back. And there's just showing what it does to the corn. And as I say, it's systemic. So if you had an early infection, sorry about that. If you have an early infection, all the leaves will show symptoms. Oh. If you have a later infection, only the top leaves will, the new leaves that would have developed. So if you can see how it develops. Now, this is the state of Minnesota, and these are all the counties where I found this virus. Now, it's interesting because this virus is transmitted by aphids, and there are no aphids that survive in Minnesota. They have to blow in from someplace else. I mean, we lived in Minnesota. When it gets 30 or 40 below, you don't have any aphids. And so what we did is this is the Maysdorf mosaic virus, and there's an A and a B strain. And where it survives in the country is in Johnson grass or sorghum halepense. It's a perennial sorghum. It's a noxious weed. And what we did is after we determined what time of the year this must have happened, we got the hold of the state climatologist, and we looked at what happened in that week in July. And there was a low pressure system here just into Canada, and then a high pressure system here over Ohio. And what you have is a tremendous amount of wind blowing up, and these are called geostrophic winds. The interesting thing is down here in Texas and Oklahoma and Kansas, there was a tremendous drought. And aphids don't get wings unless their host is failing. And so when they get wings, then they move on. Now, the other thing about this virus is it's called a non-persistently transmitted virus. Only the virus, it has to stay on the stylet, the stinging part of the aphid. It doesn't replicate. In some aphids and some viruses, the virus will replicate in the salivary glands, but this one doesn't. So we had a problem. Is how did this thing get here and still be able to transmit? Well, when you think about it, it's up in the air. It can't probe in anything, so it's not going to lose the virus. So to prove this, we took aphids and let them feed on plants, had them in the greenhouse, and then we put them in a box and chilled them. Or in one case, we used nitrogen, nitrous gas. And we were able to pull those aphids out every hour and transmit for 4 days. We ran out of aphids. So that was, the idea of something being non-persistent is kind of a bogus thing, but we had a difficult time getting our colleagues to believe it. So we ended up publishing this paper in the British Journal of Biology, Applied Biology. And it's interesting because the Italians have the problem of getting this virus, and they don't have any hosts, perennial hosts, but they like this idea of the aphids coming in from Egypt, where they have it. So anyway, it was an interesting endeavor. Okay, the next one is a disease called head smut of corn. Now, many of you know there's a common smut of corn, and and that actually is windblown, and it usually attacks the silks. And individual kernels will develop a gall, and in fact, in Mexico, it's a delicacy. You can eat it, as long as it isn't black. If it still stays gray. So anyway, but the head smut, on the other hand, this one is systemic, and it invades through the seedling, through the soil. So if the seed has spores on it, and you plant that seed, the thing germinates and then gets into the plant, and it can be transferred by machinery, by wind and seed. Here's what it does, and it's systemic. That means it starts out at the very bottom of the plant, and it can get all the way up to the tassel, and you can see the kind of galls that it produces. It's really devastating. Now, I knew about this disease because I went to Oregon State, and in the Pacific Northwest, they have this disease in sweet corn, and that's where they raise a lot of sweet corn seed. And so, but when I found it here, it was only in a couple of counties in north central Minnesota, out near Lake Wobegon. It's about where I figured it was. But anyway, and it happened in a farmer's field where we first found it, and it just demolished that. It was in bad shape. But what we found out was that this company that provides them field corn seeds oftentimes gives them sweet corn to plant along the outside so they can have a little something for themselves. And anyhow, that sweet corn undoubtedly was where it came in, because it came from the Pacific Northwest. And look what it does to an ear of corn. Rather than just a single kernel, the whole thing is smutted. So it's a devastating, and fortunately, it doesn't. We were able to control it by crop rotations and what have you. But the interesting thing is the Canadian government put a quarantine on Minnesota corn seed and Iowa corn seed, and what they were basing it on was spores on the seeds. They'd wash the seeds and then put them in a centrifuge and get a little pellet, look at it at 100 times magnification. And if they saw spores, it was gone. However, one of the things I found out was there was a weed in cornfields, panicum, that had a smut on it too, just like that. Same genus, but a different species, and it didn't attack corn. But when you harvest corn, you get those spores all over the seeds you're collecting. And what I did, I showed the Canadians at 10,000 times magnification with a scanning electron microscope, you couldn't tell the difference. So their test was bogus. So they finally removed that and got a nice letter from the Minnesota Department of Agriculture and the Iowa Department of Agriculture that we got their corn to be saved. So the precautions you do is to identify the fields that have this disease, and then harvest those fields first. Last rather, sorry, and then wash to disinfect the equipment, and use crop rotation. Don't plant corn back in that field, and try to find some resistant varieties. And there are some that are the field corns that are resistant. So that's where we went with that one. Now, this is my favorite disease, but I came to Virginia. Gray leaf spot. It actually was first described in Illinois in 1924, but it was not a problem because everybody plowed everything. But this organism survives in infested trash. So when we go to no-till agriculture, guess what you got laying all over the soil? Anyway, what you see here with this is they have these chlorotic flecks first when they first starts, and it has a pigment bordering it, and they have reduced sporulation at that time. But this thing produces a toxin that, well, here we go. This is where we know that it occurred back when we found it. Anyway, what happens, it produces a toxin that kills the tissue ahead of the fungus. And then the fungus goes into that dead tissue. and you can see what it does. Now, in Virginia, we have a lot of no-till agriculture, and we had a lot of gray leaf spot. And I was trying to convince the farmers that it was really costing them yield. And the reason that in the years in which they had poor rainfall, they had little gray leaf spot, but they had, yields that were so-so. But in the years in which they had good rainfall, they had this disease coming in, but they got their higher yields because they had the rainfall. So what I did, and I wasn't trying to promote using fungicides, but I put field plots out. Okay, shows. Yeah, here we go. This is the. Yeah, here we go. I put field plots out where I sprayed about every 2 weeks with a protectant fungicide, and in the ones that weren't treated, the yields were about 60 bushels, and the yields in which they were sprayed, they got close to 180 bushels per acre. So it trying to get people to realize that we got to look for resistance, and that's where the next part we did. There's the plots where you could see this is where we did our breeding work. This is along the New River in Blacksburg, or Montgomery County. And here I produced, it's over here, a gray leaf spotted corn for the growers to hand it out so they could see what they had, and talks about the life cycle. Okay, I should get back to this. What we ended up doing with another fellow, I had a graduate student, we identified about 5 quantitative trait loci that induced high levels of resistance. However, you have to get them in, all 5 of them in the same plant in order to get high levels of resistance. So we hired a fellow, I was involved in hiring him. He was a molecular biologist. And so we worked together. I scored plants, and he was able to determine, we isolated the molecular markers for these genes. So what you could do is use these molecular markers, take a line of corn, cross it, and then you get a little plant coming up, take a leaf and see whether you have the genes in there or not. If you don't, you toss it. If you do, you carry on and cross it with another one. So eventually, in about 2.5 to 3 years, you could get resistant corn. You could get all those genes in there. And so for that, we got a patent. And that's what, if you see what a patent looks like, we got one sitting on the desk here. It's pretty amazing. Anyway, I'll go on to the next. Now, this is where we come to the wheat part, which I don't have a lot of pictures, but this is my son, and a friend of ours was a pilot. We took this plane and flew over the plots that I had in Warsaw, Virginia, where I was conducting research. Because when I first came to Virginia, the average wheat yields were about 45, maybe 50 bushels in a good year. And we had a lot of diseases. Powdery mildew, leaf rust, septoria, leaf blotch. And so a group of 5 of us, a breeder, a couple of agronomists, me, an entomologist, and a weed scientist, we got together and decided, how can we make wheat grow better? And what we found, it wasn't unique, but this is the way they were doing it in Europe. You'd leave tramways through the field so that you could move equipment across without running over the wheat. But what we did is, previous to that, the way people grew wheat was they'd plant the wheat in the fall, it's winter wheat, they'd plant the wheat in the fall, and then about February, they'd dump all the nitrogen on the field. And that was it. And, it's like eating your old meal, a month's worth of meal in one sitting, And so that wasn't a good thing. So what we did, we had them stagger that planting, or that fertilizer activity, to put on some at planting, about 10 pounds, 10 or 20 pounds. And then at February, green up, put on about 60 pounds. And then at stem elongation, when you start getting the head, put on another 60 pounds. And then you got good yields. But other thing we did too was to narrow the rows of the wheat, because farmers were growing rows that were about 8 to 12 inches apart. And so there's a lot of wasted space. So even using the same number of seed per acre, we went down to 4-inch rows and put the seed in there and able to get yields that were tremendous. In the experimental plots, the untreated, we were getting, even in the 4-inch rows, we were getting maybe 60 bushels per acre. Where we did the best treatments, we got 106 to 110 bushels per acre. So the next thing we did was to get the farmers to try some of these techniques. So we formed clubs across the state and had the farmers take on some of these procedures. They didn't necessarily do all of them, but they took on those procedures, and they found they got some really good yields. And so even this last year, which was not a good year, the average yield for wheat in Virginia was 68 bushels per acre. And the farmers we'd worked with, I've worked with some that were getting 90 bushels or more per acre by doing the right things. When you have these tramways in the field, you could bring a sprayer across with a 20-foot boom on either side, and you could put on the fertilizer, you could put on whatever protectant chemicals. I developed a series of thresholds for treatment for these different fungal diseases. And so what we do, you try to keep the upper leaves free of disease, because that's where the photosynthate is going to get into the seed. And so I didn't necessarily want them to use a fungicide. Fungicides don't increase yields, they protect yields. So if you have the yield potential there, you need to protect it. That's why we had these thresholds. And so some of the farmers started using that, and so it's done pretty well. What we're trying to do is get farmers not to grow more acres, but to grow acres better. I had farmers that were going 20 miles down the road to go to different fields on their tractor. That's not a good way of farming. Anyway, these plots. This is show you how we do these things, and we would have these things planted here, and I use different treatments at different timings, and that's why I said I got the yields that were anywhere from 60 to up to 110 bushels per acre. That right there, that's just they took out a section. Oh, this is fallow here. Yeah, just same picture, the same thing. But I had a better picture, and I couldn't find it, where it was later in the year, and you could see there were brown plots, and there were ones that were nice and golden colored. That's the ones you want. And just to show you this is what some of the rust diseases look like: stripe rust and leaf rust, and they're wind blown, and they can do a number on a susceptible line. All right, finally, we're getting to the end here. The last disease is soybean rust. That's a relatively new disease. However, it started in Brazil because China is where soybean rust comes from, but the Brazilians got soybeans and stuff from China. And so we had an outbreak in Brazil, and it made its way up South America. And eventually, when it got to Mexico, there's a number of other legumes that are hosts for soybean rust. And we knew as a matter of time it was going to get across, and it did. And here's in 2006, the red places are we found soybean rust. And here's 2007. Now, the interesting thing is the rust will survive in the southern part of the United States. Kudzu is one of the hosts. And so, and in the south, or in that lower south, kudzu doesn't freeze down like it does here. And so, even though we had some rust here, it came in so late, it was not a problem. So we then tried to get farmers, don't spray, you don't need to. But down here, these people lose yields to soybean rust if they're not using a fungicide. Well, you can protect you put a fungicide on the soybean, and and it's a systemic fungicide, so it will actually get in and take out the rust spores. But it's not cheap. But I should mention too, both the wheat work I did and even the soybean rust, the fungicides, the active ingredient was good at about 1/4 to 1/2 ounce per acre. So you're not putting whole lots of stuff on. You might be putting 20 gallons of water on when you go across that field, but you've only got a 1/2 ounce of fungicide in that. Yeah. And the nice thing about these fungicides is they only act on chitin. And chitin is what makes the cell walls of fungi. It doesn't in plants, it doesn't in animals or humans. And so they're very safe to use. See, I think that's it. You got me. I think I did it in 1/2 hour. How about that? Yeah, ask questions, yeah. Certainly.
Any questions? Kathy had one just a minute. I saw her hand go up.
Hi, in the meantime, you said they didn't have any problems because they plowed
Yeah,
at first. And then you saw this rust. Did you have them plow in the meantime while you were developing
Well,
the
this
resistance?
isn't rust. This was gray leaf spot.
Yeah, gray leaf spot.
Well, right now, I should say in Virginia, there's a real push toward conservation tillage where you leave stuff on the ground.
But in the meantime, until you could get the resistance, did you have them plow?
No, because they didn't want to plow.
Okay.
No, and there are lots of reasons environmentally why you want to leave residues on the surface. But crop rotation is very important. You don't want to plant corn back on that residue. Yeah.
See, Ben, did you have a question?
You got to make a noise.
Eric, you talked about sweet corn seed.
Yeah, that's right.
Now, like Buster, I'm a city boy.
Yeah.
I thought corn, you grow corn, you take a kernel and plant the kernel. What's a seed?
Well, it's a kernel, a seed, whatever.
Oh, so you made
No.
a kernel.
Yeah, but Ben, what you do is you don't take the seeds out of the sweet corn that you've grown. You have to. It's a hybrid, so you have to make crosses, and that's what they do where they detassel the corn, and so they have to get very specific hybrids, and that's where they grow it in the Pacific Northwest. No, it's just a seed.
And you said some seeds come with that thing.
That's right. Yeah,
That's how you get it.
yeah, exactly. And I think that's how it happened in Minnesota. How they got it was from.
Be careful who you get your seeds from.
That's right. But I mean, the other thing is having that panicum weed with the same kind of spores on it. It's not a host. It doesn't go to corn. But it's interesting. When I showed the Canadians that their thing wasn't working, the sample that I got was a herbarium species from Manitoba. This weed, and they couldn't tell the spores either.
So you had to go a lot of traveling to find all this stuff?
Oh yeah, every 36 miles. Yeah. Yeah. Okay. Sorry.
Eric, I am a city girl, but I can recognize one plant, kudzu.
Yep. That's right.
What is the purpose of kudzu? What damage does it do? And how can we control it or find a kudzu wine recipe?
Well, I'll tell you, kudzu was actually introduced into the United States for land conservation to stop erosion. And that happened probably about 75, 80 years ago. And since then, it has created a monster. Wild rose is another one that was introduced.
How do we get rid of it? Does it kill the other plants?
It shades them out. What they use to kill it is Roundup.
Can you make wine out of it?
I don't think so.
be out of your field, but talking about problems, we have a real problem with the roses here with a rosea.
Yep, that's it.
Rosea, and have you ever worked with trying to find out what's happening there?
That's usually that's transmitted when they make cuttings of the roses. It's actually systemic and in the rose. And so you got to start buying roses that are clean. And the way they do that is to do serological testing to make sure that the material they're using is free of the virus.
Get rid of them if they do have it.
Now get rid of the rose.
Yeah, thank you. We've gone from local roses to international increase in corn yields and production. To me, it's been fascinating.
Well,
Eric,
Yeah. Well, I
Thank you.
should mention another thing. One of the things that I involved with the gray leaf spot is that back in the early 90s, gray leaf spot was found in Africa for the first time. South Africa, Zimbabwe, and I spent several months there trying to help those people get things straight away. And that was really interesting, No, it was actually in South Africa, they have 11 different languages.
okay, folks, thank you. Till next time. Eric, thank you. Big hand.